Hello everyone. Today we are going to start a new chapter, chapter number 5, Molecular Basis of Inheritance. And in today's class, we are going to cover session 5.1 and its subsession 5.1.1. So, let's get started. So, basically, in today's class, we will learn about the DNA, like what is the structure, what is the central dogma theory, and everything about DNA. So, let's get started. Disclaimer. So, what is going to be today's learning objective? First, we will understand the DNA structure, the double helix structure of DNA. Today, we are going to understand that. Then, we will identify DNA components. So, today we will talk about nucleotides and all those things and identify different, different this adenine, thymine, these things, all purine, pyrimidines, these things. And the nitrogen bases, these are the different nitrogen bases. We will talk about everything. Then we will explain the DNA function in genetic information. That what is the function of our DNA when it's like about genetic information. It's about going genetic information from one progeny to other, the inheritance thing. So we will talk about what's the function of function of DNA in it. Okay. Then we will recognize the base pairing rule. So then we will recognize this base pairing rule, like how A binds with T and how G is complement to C and all those things. Then today's index is like this. So we are going to cover the DNA, which is 5.1 session of this chapter, and then we will talk about its first subsession, that is the structure of a polynucleotide chain. Okay, then we will talk about the summary, whatever we are going to learn in the whole class. Whatever we are going to learn in whole class, we will summarize it. Then there will be some NCERT based questions, followed by keynotes, and then interesting questions that you guys are gonna solve yourself. So now let's come to the chapter. So what is DNA? What is the full form of DNA? So DNA's full form is deoxyribonucleic acid. Today we will learn everything about DNA. What is a DNA? DNA is a long polymer of deoxyribonucleotides. So right now you guys will like what is deoxyribose, what is this nucleotide they are adding after that, all those things. But you will get to know each and everything in detail in upcoming slide. For now, you just understand that DNA is a long polymer of what? This deoxyribonucleotide. We will talk about each and every component in upcoming slides. So, the length is defined by the number of nucleotide pairs, which we call base pairs. So, the length of DNA, we decide like how you decide this, what is the length of the DNA. So, it is decided by how many number of nucleotide pairs and base pairs are present in that DNA. That decides the length of that DNA. This is also the characteristic of an organism. This length of DNA can also be defined as the characteristic of an organism's. For example, the bacteriophage, lambda 174, had 5,386 nucleotides. This bacteriophage lambda has 48,502 base pairs, whereas E. coli has 4.6 into 10 to the power 6 base pair, whereas human has 3.3 into 10 to the power 9 base pairs. So this is a kind of like general things we talked about this DNA. Now we will understand its each and every component. Component as in what is nucleotide, why this is called deoxyribose. Okay, we will understand each and everything, that how we calculate this length of a DNA, that how what is this base pair, how this base pair complement each other, like everything we will learn in today class. So let's go ahead. So what is the chemical structure of polynucleotides? So as I told that DNA is a long polymer of what? Polynucleotides. So we will understand first what is a nucleotide. So nucleotide is a nitrogen base, pentose sugar and phosphate group. Actually, so what is like nucleotide? Nucleotide is a molecule, okay, that serve as a basic building block of nucleic acid. That nucleic acid can be DNA or RNA. You understand? That is a molecule that serve as basic building block of nucleic acid. Okay. So now, what is like, what are the different components of this nucleotide? First component is this nitrogenous base. Now, this what is this nitrogenous base? These are of two types, purines and pyrimidines, and that we will talk in upcoming slide. But right now, you guys should know that this component of a nucleotide, which is the basic building block of any nucleic acid, that could be DNA or RNA, they have three component, and one of the component is nitrogenous base. Then they have pentose sugar. Okay. Now, so in case of RNA, it is ribose sugar, whereas in case of DNA, it is deoxyribose sugar. Why? Because in this DNA, they don't have one OH group, that we also talk in upcoming slide. So, and then third, they have a phosphate group. So from this slide, you guys should know that what is a nucleotide? That nucleotide is a molecule that serve as a basic building block of nucleic acid. Now, this nucleotide is composed of three components. Okay, first is the nitrogenous base, second is the pentose sugar. In case of RNA, it is ribose, and in case of DNA, it is deoxyribose. And then they had a phosphate group. Now, for every component, we will talk about each of the component in detail in upcoming slides. Okay. Here, you guys just need to remember the components of nucleotide and what actually a nucleotide is. That is the basic building block of any nucleic acids. Now, as I told, we will talk about each and every component of this nucleotide. So first we will talk about the type of nitrogenous base. And from this portion of today's class, you can answer NCERT exercise question number 1. So nitrogenous base is of two type. First is purine. Okay, and purine has this two nitrogenous base. One is adenine and second is guanine. And second type is pyrimidines. These pyrimidines are cytosine, uracil and thymine. Cytosine is common to both DNA and RNA. Means like in pyrimidine, we have this cytosine, uracil and thymine. But this cytosine and uracil, this is for RNA, and cytosine and thymine is for DNA. So what is the common between DNA and RNA? This cytosine. The cytosine is the common one, but for DNA we use thymine, and for RNA it is uracil. Thymine is found in DNA while uracil replaces it in RNA. These are the structure of nitrogen bases. This is adenine, this is guanine, this is cytosine, this is uracil, this is thymine. These are our nitrogen bases. You guys don't need to remember the structure of them, but you guys need to remember like which purine will bind, complements with which pyrimidine and all those things that we also gonna talk in detail. Now, we talked about this nitrogenous base, that we have two type of nitrogenous base. First is purine, then this pyrimidine. Okay, purine is adenine, guanine, both common in DNA and RNA, whereas in pyrimidine it is cytosine, thymine and uracil. Cytosine is the common one, whereas the thymine is found in DNA, whereas uracil is found in RNA. Then it is pentose sugar. As I told, this nucleotide consists of three components. First is nitrogenous base. We talked about it. Now we come to second component, which is what? Pentose sugar. What is a pentose sugar? As the name suggests, pentose, it is 5 carbon compound named as ribose sugar. This is like, this compound is made of 5 carbon. It is a ring compound. For DNA, it is pentose sugar is devoid of one OH atom at 2' carbon position. As I told that for RNA it is ribose sugar, whereas for DNA it is deoxyribose sugar. Why it is called deoxyribose sugar? Because at the 2' carbon position, okay, of this sugar, there is devoid of one OH atom. Means there is no OH atom at the 2' carbon position. That's why this DNA, we call it as deoxyribose. Thus it named as deoxyribose, devoid of one OH atom at 2' carbon position. But for RNA, it is ribose only, because there is no such OH atom absent on this 2' carbon position. There is a presence of OH atom at the 2' carbon position. So this is how the pentose sugar look. For ribose, you guys can see at the 2' for this 2', okay, there is a presence of OH group. But if you guys will see at this deoxyribose, 2-deoxyribose, there is no OH group present at 2' carbon position. So this is the difference between the pentose sugar we are talking about. So as we have now seen what nitrogenous base and this pentose sugar, now it's time to see about phosphate group. So now we will move to the third component, which is the phosphate group. So it comes from phosphoric acid, it carries negative charge, and see, this is the structure of phosphoric acid. Okay, so this phosphate groups comes from this phosphoric acid only, and we also always says that DNA is negatively charged because of this phosphate group. So this is this phosphate group. So this is a phosphoric acid through which this phosphate group has been derived, and it carries negative charge. So this is about phosphate group, which is the third component of the nucleotide. So right till now what we have studied, So we have studied that this nucleotide is the basic building block of nucleic acid. It has 3 components. First is the nitrogenous base, which can be purine, that is adenine and guanine, and pyrimidine, which is cytosine, and thymine for DNA, and cytosine and uracil for RNA. Then we have studied about what? Then we have studied about this pentose sugar. So this pentose sugar, it's ribose for RNA and it is deoxyribose for DNA. Why? Because there is absence of OH group at 2' carbon position in DNA. Then third and the last component is phosphate group. It is what? It is comes from phosphoric acid, and this is something which is going to give the negative charge. So these are the 3 components of the nucleotide, and this long polymer of this polynucleotide. What it makes? It makes DNA. Now, formation of nucleosides and nucleotides. So now what is this nucleoside and nucleotides? So you guys pay attention, because this is something confusing. People can get confused in it. So what is this nucleotide which you are talking about? Is nucleoside plus phosphate group. So here you understand like this. Nucleoside is what? Nucleoside, as it's its side. Okay, so here you can see it is, what is nucleoside is? It is base, the nitrogenous base, and the sugar, that is the pentose sugar. Okay, these 2 are called nucleoside. Okay, and you know how I used to remember this? So when I was in 11th, 12th, I used to remember it like that it's side means S, and S means second means 2. So nucleoside is base plus, that is nitrogenous plus sugar. And how it is linked together? So here you can see this is sugar and this is nitrogenous base. These 2 are making our nucleoside. So How they are linked together? They are linked together via N-glycosidic linkage. What is a N-glycosidic linkage? So for your knowledge, I will tell you that N-glycosidic linkage, it is a covalent bond only, and it is between the anomeric carbon of sugar and the nitrogen atom of the nitrogenous base. Now you can ask, what is the anomeric carbon of this sugar? Nitrogen of nitrogenous base, you can understand that is the N of the nitrogenous base. Now what is the anomeric carbon? Carbon that's the part of carbonyl group in sugar open chain form. So the carbon which is the part of carbonyl group in this sugar open chain form is the called anomeric carbon. So this is the link, covalent link between these two. Now, as I told you guys can remember it in this form, that nucleoside, S means second and second means 2. So base, nitrogenous base, and sugar, this pentose sugar, is going to give us the nucleoside. And the linkage which they form in between is the N-glycosidic linkage. Whereas now, this sugar and base, what we call nucleoside, when combined with this phosphate group via phosphoester linkage, is called nucleotide. And what is nucleotide? We have studied that this is the basic building block of nucleic acid. So now you guys can relate that what is this nucleoside and nucleotide, and tide means 3 and 3 means T means 3 and 3 means 3 components, this nucleoside and phosphate group. Now you guys can ask that what is this phosphoester linkage? So the phosphoester linkage is nothing but the linkage between the phosphate group and OH, hydroxyl group of the sugar. When they combine together, this is called phosphoester linkage. So now I think you guys understand that what is this nucleoside and what is this nucleotide. Nucleoside is just composed of pentose sugar and nitrogenous base via N-glycosidic linkage, whereas when in this nucleoside there is an addition of phosphate group via phosphoester linkage, this is what we call whole a nucleotide. And this nucleotide is what? It is the basic building block of any nucleic acid that we have studied in detail, that what are the 3 components and all those things, how they differ in DNA and RNA and everything. Now next, okay, so now the key linkages, that is the N-glycosidic linkage, as I said, joins the nitrogenous base, as I said earlier also, to the 1' carbon of the pentose sugar, which is forming a nucleoside. So as you guys can understand that this is a nucleoside, why? That there is a sugar, deoxyribose sugar, and then there is a nitrogenous base, and this is a cytosine. Okay, so what happened? This nitrogenous base is attached to this 1' carbon of pentose sugar. We number it like this now: 1, 2, 1 dash, 2 dash, 3, 4, 5, this dash. So this is at the 1 prime it is combined at the 1 prime or 1' carbon. So this is how N-glycosidic bond are formed in the nucleoside. Whereas what is the phosphoester linkage? It joins a phosphate group to the 5' OH of the nucleoside, forming a what? A nucleotide or deoxynucleotide. Nucleotide in the case of RNA and deoxynucleotide in the form of when it's DNA. So here you guys can see that this is a ribonucleotide, okay, and this is the glycosidic linkage where this nitrogenous base is binding to this carbon. But here you guys can see at the 5' OH group of nucleoside, there has been the joining of a phosphate group, and this is the phosphoester linkage. So these are the phosphoester and phosphodiester bond. So now what is more important here is that we will now study this 5' to 3' to 5' phosphodiester linkage and how it is joining the 2 nucleotides. So this 5' to 3' 5' phosphodiester linkage, what it is doing, it is joining the two nucleotides. The 3' carbon of one sugar is linked to the 5' carbon of next sugar via a phosphate group. So as I told that this DNA and all those things, but it is the long chain of deoxyribonucleotide. So now how they are joined together? So the 3' carbon of one sugar, okay, is linked to 5' carbon of next sugar. How? Via this phosphate group. This form a dinucleotide. When two nucleotide will join like this, it will form a dinucleotide. But when more number of nucleotides start joining like this, it will make a polynucleotide chain. So here you guys can see that what it is happening. So there has been joining of, here you guys can see this is one of the deoxynucleotide. Okay, so now what's happening at this 5' prime or 5' carbon, okay, is attached to this 3' carbon of the different nucleotide, via this phosphate group. And this linkage, this whole linkage is we call 5' to 3' to 5' phosphodiester bond. So like this, 2 nucleotide, 3, 4, 5, 6, and then the whole number, a very large number of nucleotides bind together and form a polynucleotide chain. So this is how this polynucleotide chain has been made, and this is the 3' to 5' phosphodiester linkage. This you guys need to understand very properly. This is what? This is the 5' and 5' one carbon, okay, is attached to this phosphate, and this phosphate is now attached to the 3' carbon of the next nucleotide. Like this also, then how it will happen, then 5' of here will be linked to the 3' of this next carbon, and like this, they will make a very long chain and make a polynucleotide chain. Okay. Now, till now we have seen the, like, what is this nucleotide, what is the nucleoside, what is like what are the different components of nucleotide, how two nucleotide join to become dinucleotide. Then, like, if more number of nucleotide will come with this 3' to 5' phosphodiester linkage, we can make a whole polynucleotide. And what is a long chain of this deoxyribonucleotide is a DNA. So now where we will come after that, we will see some historical content, like how these discoveries has been made and what are these discoveries. So first, Friedrich Mischer in 1869 identified DNA and called it nuclein. So in 1869, there was a scientist named Friedrich Mischer, and what he did, he identified DNA and named it what? He named it as nuclein. What was its structure elucidation? Like how the structure of this DNA has been known and all those things. James Watson and Francis Crick gave the structure of DNA in 1953. So Watson and Crick, they did what? They gave the structure of the DNA. They proposed a very simple but famous double helix model for the structure of DNA. So this double helix model of DNA. So Watson and Crick are the people who has given this structure of DNA. We will study what they have told and everything in upcoming slides. It was based on what? It was based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin. So this model they proposed, okay, so how they proposed it? So they proposed by the data which they have gotten from X-ray diffraction, and this X-ray diffraction data is produced by whom? These are produced by Maurice Wilkins and Rosalind Franklin. Now after this, if Watson and Crick has given the double helix structure of DNA, Chargaff's rule. From this you can answer the NCERT exercise question number 2. What was the Chargaff rules? This is one of the very important discovery also that he proposed what? He proposed this. He proposed an equation. Let me tell you. Huh. So Edwin Chargaff, he proposed this ratio thing, this formula thing for double standard DNA. So what this ratio means, what this all about is that the ratio of A plus G by T plus C is always 1. Now A plus G, what is A and G? A and G is the purine, and what is T and C? They are pyrimidine. So this is the when we will add this whole purine and divide by this pyrimidine whole, this ratio will always 1. Why it is so? Because ratio of adenine to thymine and guanine to cytosine are equal to 1. That is A equals to T and G equals to C. Why they are saying that the ratio of adenine to thymine and guanine to cytosine are equal to 1? Why? Because A binds with T, they complement each other, okay? And A, with the help of 2 hydrogen bonds, bind with T. Means if there are 5 number of A present in the DNA helix, then there will be definitely only 5 number of T has been present in that helix. If there are 20 G in that DNA helix, then there will be 20 cytosine only in that DNA helix. That's why they say that the ratio means if we will divide adenine by thymine, it will be 1 because both are same. Okay, if you will divide 5 by 5, it will be 1. If you will divide 30 by 30, it will be 1. So he's saying that the adenine to thymine ratio as well as the guanine to cytosine ratio is always equal to 1. Because why? Because A equals to T, because we know that A is going to bind to T with the help of 2 hydrogen bonds, and G is bind to C with the help of 3 hydrogen bonds. So they are equal. Okay, so the ratio will be equal, and if we will calculate this whole purine concentration and all by pyrimidine, it will also be always 1. We will see this in one of the example also, numerical example, I think, which is in the NCERT based question. Now, after these rule, this we have seen this all Chargaff's rule. Now we will talk about the DNA double helix structure which Watson and Crick has given in 1953. What are the features of those DNA double helix structure? From here you guys pay more attention because this is very important. So, what are the different features of that DNA double helix? Let's see. We will see one by one. First, this DNA double helix is made up of 2 polynucleotide chain. We have studied that what is this polynucleotide chain, how they are formed with that 3' to 5' phosphodiester linkage and all those things we talk about nucleotides. So till now we know that what is polynucleotide chain. What this salient feature of DNA double helix is that this DNA helix is made up of 2 polynucleotide chain. There has been 2 polynucleotide chain in that. DNA consists of 2 polynucleotide chain. Backbone of the DNA is composed of what? Sugar and phosphate. Whereas the nitrogen bases always remain projects to inwards. Its bases remain project to inwards. So here you guys can see this is an NCERT figure only, that this double stranded polynucleotide chain is DNA double helix DNA structure is made up of 2 polynucleotide chain. One is this and 2 is this. This phosphate and sugar is the backbone of this DNA. Okay, but this adenine, this nitrogen bases, that is adenine, thymine, guanine and cytosine, they remain projects to inward. Why? They want to have this, they make a complementary bond with each other with the help of hydrogen bonds. So they will be like inwards. And whereas the sugar and phosphate is what? They are the backbone of the DNA. So this is the first salient feature of this DNA double helix, that they are consist of 2 polynucleotide chain. Let's see in second. Now, what is the polarity of that? They are anti-parallel. 2 polynucleotide chain have anti-parallel polarity. Means if one is from 5 dash to 3 dash, other will be from 3 dash to 5 dash. Okay. One chain runs, as I said, 5 dash to 3 dash, whereas the other runs from 3 dash to 5 dash. You guys can see, like, the one chain is what? This one chain is what? This is from 5 dash to here, 3 dash, whereas the other is 3 dash to 5 dash. Okay, they are in the opposite direction. They have this anti-parallel polarity. Now, so what we have studied, that they are of the 2 polynucleotide chain. Okay, then we have studied that they are antipolar, means one if it's running from 5 dash to 3 dash, other will be from 3 dash to 5 dash. Now third is that they are having the complementary base pairing. From this you can answer this NCERT exercise question number 3. What is this complementary base pairing? It means that adenine pairs with thymine via 2 hydrogen bonds, and guanine pairs with cytosine via 3 hydrogen bonds. Means purine pairs with pyrimidines, because we know that as adenine and guanine is purines and thymine and cytosine is what? Is pyrimidine. In case of RNA, it will be uracil then rather than thymine. Now, purine always pairs with a pyrimidine, making a uniform helix width. Means purine is going to pair with pyrimidine only. Why? Because they have to make a uniform helix width. Means I'll show in the figure. So it's not showing it here. Like I'll show in the other figure, that if they're making a helix, so the width of the helix should be uniform. And how it is possible? If they make a pairing with purines, make a pairing with pyrimidine. So here you guys have seen in last figure also that this phosphate and sugar, this is making backbone, and this nitrogenous base is inverts, and this adenine and thymine are complementary to each other, and they are pairing together via 2 hydrogen bonds, whereas this guanine and cytosine is pairing with each other via 3 hydrogen bonds. Now next feature, that they are right-handed helix. 2 chain of DNA coiled in a right-handed direction. So their direction is right-handed. Pitch is 3.4 nanometer per turn, which are also 34 angstrom. So now in this you guys need to understand that what is a pitch. So pitch is what? It is the distance. Okay, it is the distance along the helix axis for one complete turn of the DNA helix. So it is the distance along the helix axis for what? For a complete one turn. Okay, so this is the whole one turn that this DNA will take. So the distance between that is what we call pitch. So what is this distance along the helix axis for one complete turn of this DNA helix? Is 3.4 nanometer or 34 angstrom. You guys remember 3.4 nanometer only. In previous slide I told you that the width will remain uniform because the purine going to bind complementary with pyrimidine. So you guys can see that purine will always complements pyrimidine, and there will be a uniform width between this double helix structure. And yeah, so the pitch is what? The pitch is 3.4 nanometer, and what it is the one complete turn of the DNA helix, the distance along the helix axis for one complete turn. Okay, so this is what we call pitch. Now in this whole one turn, there is 10 base pair. Okay, we guys can even count 1 2 3 4 5 6 7 8 9 10. So here it might be like height between in that. So in one turn, which is a distance of 3.4 nanometer, there are 10 base pairs. So now, can we calculate the distance between 2 base pairs? Yes, we can. Why? Because we are saying that the one complete turn has a distance what? 34 angstrom or 3.4 nanometer. So if it's like 3.4 nanometer for 10 base pairs, okay, so for one base pairs it will be 3.4 divided by 10, and how much it will be? It will be 0.34 nanometer. So the distance between two base pairs is what? It is 0.34 nanometer. As I said, this is the right-handed helix. It is like its direction is in right hand. Okay, now as I said, approximately there has been 10 base pairs per turn. So what will be the distance between base pairs? Obviously 0.34 nanometer. Why? Because for 10 base pairs it is 3.4 nanometer. So for one base pair it will be like what? 3.4 nanometer by 10. So as I said, now what are the other feature? So the other feature is base stacking. Base pairs stack on top of each other, that we can see in the, we have seen in the other previous pictures also, that the base pairs are stacking on each other, on top of each other, and this is adding what? It is adding the stability to the helix in addition to the H bonds. So first of all, this nitrogen and base, they are complementary to each other, and they are base pairing with the help of this H bonds. This is adding stability to the helix. Along with that, the base pairs are stuck on top of each other, and that's why they are giving more, they're giving more stability. Now comes central dogma of molecular biology. What is the central dogma of molecular biology? This is something which proposed by Francis Crick. What he has proposed, we will see. It describes the flow of genetic information in a biological system. So this central dogma describes that how the genetic information has been transcribed and translated. Means what is the flow of this genetic information? Genetic information flows from DNA to RNA and then RNA to protein. Means our genetic information is all contained in this DNA. Then it's transcribed, it goes into this RNA, and then at last it is translated as protein. So, whereas in some viruses, the flow of information in reverse direction, means the flow, like in other cases, in most of the cases, it is like from DNA to RNA. But in the cases of virus, the flow of information is what? It is reverse, that it is from RNA to DNA. So here you guys can see the central dogma thing. You can see here that this DNA is first transcribed into this mRNA, that is messenger RNA, and this messenger RNA is then last translate into protein. And what is this replication showing? We know that the DNA replicates itself and all those things, but here is the central dogma that DNA transcribe into messenger RNA, and this messenger RNA then translate into what? Into protein. And this protein is like then responsible for all the functions a body performs. Transcription. What is transcription? It is the process of making RNA from DNA template. What is then translation? It is the process of synthesizing a protein from the information on an mRNA molecules. So what is transcription? Transcription is a normal process in which the in which the body makes RNA from what? From the DNA template. Okay, and what is this translation? So the translation is the process in which this protein is synthesized. From where it synthesized? From the information that is on the mRNA molecule. So from the information on that mRNA molecule, there was like genetic code and all those things. This protein has been synthesized. So this is what transcription and translation means. Now, what is reverse transcription? In some viruses, such as retroviruses, the flow of information is in reverse direction. How it is in reverse direction? That, as I told, that in normal transcription, what we do, we make an RNA from DNA template. But in reverse transcription, what we do, we make DNA from RNA template. And we have talked about this retrovirus in this biotechnology chapter and all, and that how they have, like, it's in the reverse direction. But you guys just know this, that in viruses, example retroviruses, the flow of information is in reverse direction, that it will go from RNA to DNA. So what is the summary of all the thing today we have learned is that DNA, that is deoxyribonucleic acid, is the genetic material present in all living organisms. It is a double helix structure discovered by Watson and Crick in 1953. Watson and Crick has discovered that DNA is what? It is a double helix structure, and we have learned its salient feature also, that they are anti-parallel and all those things, that the one turn is of 3.4 nanometer and the distance between the base pair is 0.34 nanometer, and this adenine binds with thymine and guanine binds with cytosine with the help of 2 and 3 hydrogen bond respectively. They base pairs stack over on the top of each other to make it more stable. This is the double helix structure, which is described and discovered by Watson and Crick in 1953. Then we have 4 nucleotide bases: adenine, thymine, nitrogenous bases, that is the adenine, thymine, guanine, and cytosine. It is a double helix. Now talking about base pairs, A pairs with T, G pairs with C. A pairs with T with the help of 2 hydrogen bonds, whereas G pairs with C with the help of 3 hydrogen bonds. It carries genetic information and instruction for growth, development and reproduction. Located in the cell nucleus, nuclear DNA, and mitochondria also has its DNA, that is mitochondrial DNA. So the backbone of DNA is sugar and phosphate with attached bases, and they are inward, and the face is inwards, and we know that they complement each other, this purine and pyrimidine. And all purine will always bind with pyrimidine, so that there has been a stable helix width, uniform helix width. So this is all about today's summary. Now let's solve some NCERT based questions. Okay, so first question says, which of the following is not a component of a nucleotide? So they have given us 4 options and they are asking us which among them is not a component of nucleotide. So first of all, what we have learned, what is a nucleotide? Nucleotide is the basic building block of the nucleic acid. Okay, that could be DNA and RNA. Then we have studied that it has 3 components. So what are those components? First was the nitrogenous base, which was purine and pyrimidine. Then it was pentose sugar, which was ribose and deoxyribose. And third was the phosphate group. So what is going to be answer of this question? So the answer of this question is going to be amino acid. Nucleotides are composed of a nitrogenous base, a pentose sugar, and a phosphate group. Amino acid are the building block of proteins, not nucleic acid. So this is a whole figure of a nucleotide. This is the phosphate group, this is pentose sugar, and this is the nitrogenous base. Now second question. So I told you that Chargaff's rule we will understand more nicely with the help of one question. So let's see the question. So the question is, in double stranded DNA molecule. Okay, so there is a double stranded DNA molecule. If the amount of adenine is 20%, what is the approximate percentage of guanine? Options are 20%, 30%, 40%, and 60%. So the question is saying, so they have 20% of adenine. Okay, this double stranded DNA molecule have 20% of the adenine. So now tell us what is the percentage of guanine. So here you guys understand thing. Okay, what you guys need to understand, that adenine is like how much percent? It is 20%. So thymine, as we know that adenine is going to complement, going to base pair with thymine only, and we know that A equals to T. So now if adenine is 20, then thymine will be also 20. Now we know that whole purine by pyrimidine is equals to 1. Okay, so now we know that this adenine is 20 and this thymine is 20. It means 40% thymine and adenine is taking. So now what will happen? How much percentage is left? 100% is left, and 40 has been taken by adenine and thymine. So now if you will minus 100 minus 40, what will be left? 60. So now 60% is something that is guanine and cytosine is going to occupy. Now we also know that guanine is equals to cytosine, and if 60% is left, then it will be like divided how? 30% will be divided to guanine and 30% will be divided to cytosine. So this is will be the answer will be 30. Let's see the answer. Yeah, the answer is what? 30%. I will make you understand it again. A is 20%, then we know that thymine also going to be 80%, 20%. A is 20%, then thymine is also going to be 20% because we know that A equals to T. Now, if A plus T equals to 40%, then how much will be left? Remaining will be obviously G plus C. So now G plus C is going to be 100 minus 40. So 100 minus 40 is what? 60. So G plus C equals to going to be 60%. Now we also know that G equals to C. So now if you will put like G, we will substitute G here as C, then it will be like 2C equals to 60. So 1C will be 30%. And if C is 30%, G will be also 30%. So this thing you guys need to understand. Now next question says us, which linkage joins a nitrogenous base to 1' carbon of the pentose sugar in a nucleoside? So they are asking about this now linkage. So which linkage is going to join the 1' carbon of the pentose sugar to the nitrogenous base in a nucleoside? Is it phosphoester linkage? It is N-glycosidic linkage. It is phosphodiester linkage, or it is hydrogen bond. So here like there is a like hint you guys can get that definitely nitrogenous base, they are going to be like from N. So you guys can link like that, like N-glycosidic linkage. But let's see the answer. Answer is N-glycosidic linkage. It specifically connect what? The nitrogenous base to the 1' carbon of the sugar, whereas the phosphoenter linkage connects the phosphate to the sugar, and the phosphodiester linkage connects to nucleotide, that 3' to 5' we talked about, whereas the hydrogen bonds do what? They hold base pairs together in a double helix. So this is the whole thing. This is the nitrogenous base and this sugar holding together by glycosidic bond. Then this is a phosphoester bond, and if there will be more than two nucleotides together, then there will be 3' to 5' phosphodiester bond linkage. Next question. Now next question is what? Next question is, what is the approximate pitch of the DNA double helix according to the Watson and Crick model? So they are asking about the approximately pitch. Pitch means what? The single turn, the distance along the helix axis for one complete turn of the DNA helix. So what is this? Is it 0.34 nanometer? Is it 3.4 nanometer? Is it 34 nanometer? Or it is 5.386 nanometer? Answer is 3.4 nanometer. The provided text stated that pitch of the helix is 3.4 nanometer. It is so. How is this saying 0.34 nanometer is a distance between two base pairs? Because we know that what we know? We know that in a single turn there will be approx 10 base pair, and if the distance is 3.4 nanometer and in that distance 10 base pairs are there, then the distance between one base pair will be 3.4 nanometer divided by 10, and it will come out to be 0.34 nanometer. That's why it is saying that 0.34 nanometer is the distance between base pairs. So this is the same diagram which we have already studied in today's class, that this is the distance, this is the whole pitch, and this is the distance between two base pairs. So the flow of genetic, next question says ask, the flow of genetic information from RNA to DNA observed in some viruses is known as, we know that the normal central dogma is what? When the genetic information is from DNA to RNA to protein, DNA to mRNA to protein, whereas the genetic flow in some viruses is RNA to DNA, and what it is known as? So it is known as reverse transcription, because from conversion of this DNA to RNA is known as transcription, so the reverse will be known as reverse transcription, and it has been seen in some retrovirus. So what I said is same, that central dogma typically is flows from DNA to RNA to protein, whereas the reverse transcription is the specific process where RNA acts as a template to synthesize what? To synthesize DNA. So this is the normal central dogma, and the reverse will be reverse transcription, and it is shown in some retrovirus. Now next question says us, which of the following statement about DNA double helix is incorrect? They have given us 4 statements and they are saying us we have to tell them which is incorrect when it comes to DNA double helix. First option says that the two polynucleotide chain have anti-parallel polarity. We know that the two polynucleotide chain have an anti-parallel polarity. If one is running from 5' to 3', other will run from 3' to 5'. So first is correct. Let's see the second. Adenine pairs with thymine via 3 hydrogen bonds. We know that the adenine pairs with thymine, but is it via 3 hydrogen bonds? No, it is via 2 hydrogen bonds. So this statement is incorrect, but we will see the other options too. The 2 chains are coiled in a right-handed fashion. Yes, we know that the 2 chains are coiled in the right-handed direction, so it is right. Fourth, guanine pairs with cytosine via 3 hydrogen bond. Yeah, it is correct that the guanine pairs with cytosine with the help of 3 hydrogen bonds. So the answer is, means the incorrect statement, but the correct answer is B, that adenine pairs with thymine via 3 hydrogen bonds. Adenine pairs with thymine via 2 hydrogen bonds, not 3. Guanine pairs with cytosine via 3 hydrogen bonds. The other statements are correct feature of the DNA double helix, and this is the double-stranded polynucleotide chain which we have seen and discussed in the previous slides. So what are today's keynotes? DNA full length in one human cell is about 6 feet. It's too much. 99.9% of DNA is identical between humans. We share 98% of DNA similarity with chimpanzees, 50% with bananas, and DNA can store 250 petabytes in a single gram. It takes 30 minutes to unwind the DNA, and DNA was first discovered in 1869. Every human has a unique DNA. In spite of this 99.9% of DNA similarity, they are identical. Every human has a unique DNA. So there are some interesting questions that you guys gonna solve yourself. So one of the question is, considering the vast difference in the DNA length between a human and E. coli, what are the biggest challenges in accurately replicating such massive genetic material? Second, beyond carrying genetic information, how might the sugar phosphate backbone's consistent structure contribute to DNA overall stability and function? Third question says, if Chargaff rules didn't apply, example A didn't equal to T, what fundamental aspect of DNA double helix structure would be compromised? So these are the things which we have talked, like not direct but indirectly we have like almost answered the every question. So now you guys need to search it yourself and write it in your notebooks. Thank you. I will open this DPP questions. So the first question is, which of the following nitrogenous base is present in RNA instead of thymine? Means which nitrogenous base is present in RNA instead of thymine. So we know that in this case of pyrimidines, we have uracil instead of thymine in RNA. Second question says, in the DNA double helix, the two polynucleotide strands are. So in a DNA double helix, the two polynucleotide strands are like what? Are they parallel with same polarity? No. They are anti-parallel with complementary base pairing. Means if we will run, it is like 5' to 3' polynucleotide, then other will be 3' to 5'. Are they coiled in left-handed fashion? No, they are coiled in right-handed fashion. Linked via phosphodiester bond between bases? No. This phosphodiester bond, they are linked between two nucleotides, whereas this in between the bases, they are linked via hydrogen bonds. So the answer is second, that they are anti-parallel with complementary base pairing. Third question says, the distance between two consecutive base pairing in a DNA helix is approximately. So we know that the distance between two consecutive base pair is 0.34 nanometer. Why? Because in one turn where there are 10 base pairs, the pitch is 3.4 nanometer, and in one pitch there are 10 base pairs. So if we will divide 3.4 nanometer by 10, we will understand that the distance between the two consecutive DNA base pair is 0.34 nanometer. Next question, save and next. Which structural unit of chromatid consists of DNA wrapped around histone octamers? So they are asking us this, which structural unit of chromatin is consist of DNA wrapped around histone octamers. So we know that nucleosomes have this 200 base pairs, that's been DNA base pairs which has wrapped around histone octamer. This we are going to study more in upcoming classes. Now, last question. If the length of E. coli DNA is 1.36 millimeter, how many base pair does it approximately contain? So this question is like they have given the length of the E. coli DNA, which is 1.36 millimeter, and they are asking us that how many base pair does it approximately contain. So they are asking us the means number of the base pair they approximately contain. So we hear from node, so we have learned that. So how we can calculate the length of any DNA? So we can calculate the length of the DNA by number of the base pair they do have into distance between that base pairs. So we know that length here is given as 1.636 millimeter. So if you guys want to convert it in millimeter into meter, you will guys understand that it will be 1.36 into 10 to the power minus 3 millimeter. Meter, sorry. So the length will be how much? 1.36 into 10 to the power minus 3 millimeter. Now, as I said, that length will be equal to number of the base pairs into distance between the base pairs. And what we have studied, and even in previous question it has been asked, that what is the distance between two consecutive base pair? That is 0.34 nanometer. And if you guys will convert 0.34 nanometer into meter, it will be like 0.34 into 10 to the power minus 9 meter. Now what you will do? Length divided by distance between the base pair will give you number of base pairs. So length will be here 1.36 into 10 to the power minus 3 meter divided by 0.34 it will be 0.34 into 10 to the power minus 9 meter. So if you will take this 10 to the power minus 9 above in numerator, it will be like 10 to the power minus 3 into 10 to the power 9. So it will be like, you calculate like minus 3 plus 9 equals to 10 to the power 6. And now if you will calculate 1.36 divided by 0.34, it will come 0.34, it will come 4. So the answer will be 4 into 10 to the power 6. So let's see, we will mark 4 into 10 to the power 6. So we will save this. Now we will submit today's test, and yes, we want to submit it, and we can see all of our answers are correct. So thank you. Is it all for today's class?